In this session, I am taking up enzymes, definition and classification. Objectives set for this session are: at the end of the session, the learner shall be able to define enzymes and classify enzymes. Before taking up these two issues, let us have some brief idea about the enzymes. We know the word catalyst. We also know catalysts accelerate chemical reaction. Though catalyst participates in a reaction, but there will not be any chemical or physical changes in the catalyst. Only the changes will be of the substrate which on catalyst is acting. Enzymes are known as biocatalysts. Biocatalysts are so much superior than the catalysts of either inorganic or metal nature because they are highly specific. Okay, so biocatalysts carry out biological reactions, catalyze the biological reactions. They increase the rate of any biological reaction. As I said, they are highly specific. So they have the sensitivity. The small amount can carry out convert the large substrate into a product. Okay, so now let us take up the definition, how we can define enzymes. So the first objective is definition. Definition of enzymes. The enzymes are soluble, okay, colloidal, being proteins, colloidal, organic, catalysts formed by the living cells. Living cells formed by the living cells. Then specific in action and mostly protein in nature, chemically they are proteins, except some enzymes which are ribozymes. RNA is the chemical structure. Okay, they are inactive, mostly inactive at 0 degrees and destroyed at destroyed at higher temperature. Suppose it is 100 degrees centigrade. Let us take up the second objective, that is classification. Classification of enzymes. They are classified into 7 classes. The last one is recently added one. Okay, so they are 7 classes. Okay, that is a 1, 2, 3, 4, 5, 6, 7. This classification is described by Enzyme Commission of International Union of Biochemistry. Okay, the first one is, as I said, EC1, then EC2, then EC3, EC4, EC5, EC6, EC7. EC means Enzyme Commission. So the first one, the group is oxidoreductases. Oxidoreductases. The second one is transferases. Transferases. The third one is hydrolases. The fourth one is ligases. Okay. The fifth one is isomerases. The sixth one is ligases. The seventh one is translocases. Translocases. So they are the 7 classes of enzymes. 7 classes of enzymes. Okay. Now, we'll take up one by one. The first one is EC1. What is EC1? Oxidoreductases. It is oxidoreductases. What they do, catalyze oxidation reduction reactions. Okay, reactions. Let me show. Suppose this is the one which is reduced. Okay, then the next one is here, it is oxidized. Okay, this is reduced, this is oxidized. By the action of oxidoreductases, what will become? This will become oxidized. This will become oxidized and this will become reduced. Okay, oxidation reduction reactions are taking place by the action of oxidoreductases enzymes. So let me quote one particular reaction. The reaction which all we know is taking the lactic acid. Okay, lactic acid. This is the lactic acid. And plus coenzyme NAD+. This is lactate. This is NAD+. Okay, then what happens? By the action of enzyme. What is the enzyme? LDH. So oxidation of lactate is taking place, converting into pyruvate. This is called pyruvate, and NAD is converted to NADH plus H+. So here what happens? These hydroses, they are removed and they are shifted to this place. Lactate is converted to pyruvate, NAD plus is converted to NADH plus H+. What are the various enzymes under this group? Various subclasses are there. They are dehydrogenases. Okay, reductases, then oxidases, then peroxidases. The examples of enzymes under these various subclasses are LDH, G6PD, part of HM pathway, then peroxidase, okay, then cytochrome oxidase, okay, and maybe malate dehydrogenase. Let us take up the second one, that is EC2. What are these enzymes? They are transferases. Let us see. Here there is a group attached to one substrate. Then what happens? Here, this group is transferred to this one. It is not hydrogen, other than the hydrogen. Okay, transport to other substances. Okay, then let us see here. Because of these transferases, A plus. Okay, then so these transferases, they transfer the groups, catalyze the transfer of C, N, or okay, P, or methyl groups from one substrate to another substrate. Okay, then so let us take an example. We'll take amino acid serine. Okay, amino acid serine. Serine is the amino acid. That amino acid we will take up. So it will be transferred to tetrahydrofolate. So by the action of enzymes, serine hydroxymethyl transferase leads to formation of glycine plus 5,10 methylene tetrahydrofolate. Okay, 5-10 methylene tetrahydrofolate. This is serine. This is tetrahydrofolate, serine hydroxymethyl transferase. So leads to formation of glycine. Then the 5-10 methylene tetrahydrofolate. Okay, so this is how the transfer is taking place. There are important subclasses are there. Aminotransferases. Aminotransferases, or we can say transaminases. Transaminases. Okay, then kinase. Transcarboxylase. The examples are aspartate transaminase, alanine transaminase, okay, ornithine transcarbamylase, transcarbamylase, okay, then hexokinase, creatine kinase, etc. The various enzymes are there. Various important enzymes are there. Then let us take up the third one. Then that is EC3. They are hydrolases. They are hydrolases. Okay. Cleavage of bonds by addition of H2O. So bonds are cleavaged. So by addition of H2O. They are hydrolases. Let us see. Let us see. Okay. So here there is a bond is present. In the presence of H2O, so the bonds are broken and two substrates are formed. Two substrates are formed. Okay. So let us take up the urea. Down in the presence of urease enzyme. Urease enzyme. So here what happens leads to formation of ammonia and carbon dioxide by the action of urease enzyme. Okay. Then what are the various examples? What are the various examples under this group? They are acid phosphatase, acid phosphatase. Okay. This is of course urea. This is carbon dioxide and acid. Acid phosphatase, then lipase, amylase, trypsin, all proteolytic enzymes, trypsin, like other enzymes, lactase, okay, then also sucrase, okay, like that. Then ALP, alkaline phosphatase. Okay. Let us take up the next one, that EC4. What are these enzymes? Ligases. Ligases. So they catalyze cleavage of, okay, CC, CS and CN bonds. What is the difference between hydrolases and these ligases? Here there is no water involved. There is no water involved. Let us see how this reaction is taking place. Okay, there are two groups are involved. By the action of ligase, you can see a leaving behind a double bond. Leaving behind a double bond. Let us see here. Take the example, well-known example, that is pyruvate, pyruvic acid, end product of glycolysis. Pyruvate. Okay. So pyruvate, by the action of pyruvate decarboxylase, leads to formation of, okay, acetaldehyde plus carbon dioxide. This is acetaldehyde. Okay. Then various subclasses are there. So there are many are there like aldolase, an important enzyme of glycolysis, fumarase, an important enzyme of citric acid cycle. Okay, arginosuccinase, an important enzyme of urea cycle. Okay, carbonic anhydrase. Okay, decarboxylase. There are many such enzymes are there. There are many such enzymes are there. Okay, then the next one is EC5. What are EC5 enzymes? Isomerases. What are EC5 enzymes? Isomerases. Okay. So in the isomerases, catalyze rearrangement of rearrangement of optical or geometric isomers. Optical or geometric isomers. Suppose here we have the rearrangement is taking place. Okay, like that. Let us see one example. That is methylmalonyl CoA. Methylmalonyl CoA. The rearrangement, how the rearrangement is taking place. So by the action of enzyme mutase, methylmalonyl CoA mutase, the rearrangement is taking place. Okay, let us see formation of succinyl coenzyme A. It is methylmalonyl coenzyme A. It is succinyl coenzyme A. Okay, what are the various enzymes under this group? Phosphoglucomutase, okay, phosphoexose isomerase, okay, epimerase, okay, retinol isomerase, okay, these are various enzymes under this group. Let us take up sixth one. What are these enzymes? Ligases. What ligases do? Catalyze formation of okay, bonds between what? Between CO or CS or CN groups. Okay. So what happens? Coupled to hydrolysis of high energy phosphate. This reaction is coupled to hydrolysis of high energy phosphate. So along with this one, so there is ATP is converted to ADP or GTP is converted to GDP. Okay. So these are ligases. Let me show how the reaction is taking place. So A plus B has to be connected at the cost of ATP, hydrolysis. So AB plus ADP plus IP. Okay, let us see the actual reaction here. The pyruvate we are taking here. Okay, pyruvic acid. This is pyruvate. Okay, it combines with carbon dioxide. So the coupling is taking place here. So leads to formation of oxaloacetate. Okay, intermediate of citric acid cycle. Oxaloacetate. Okay. Then what are the various enzymes? Glutamine synthetase, synthetase, as you know, where ATP is used. Okay, then pyruvate carboxylase, and the most important DNA ligase. The most important DNA ligase. The final one is EC7. What are these enzymes? Translocases. Translocases enzymes. What is it? Translocases. See that a molecule is this side of membrane. It goes, an ion is going to this side. Either a molecule or ion is going this side. So by the action of translocase. So from this we can understand they catalyze the movement of molecules or ions across the membrane, or their separation within the membranes, or their separation within their membrane. Okay, what are the translocate? What are they? Translocate. Translocation of H plus H ions, hydrons, inorganic cations, anions, peptides, carbohydrates, amino acids. Let me now sum up the things which you have studied. In this session, we have taken up the definition and classification of enzymes. As I said, enzymes are biocatalysts. So they increase the biological reactions. Okay. So they are superior than the catalysts. They are superior than the catalysts. Okay. That means inorganic catalysts and metals. They are much faster, more specific, and highly sensitive. Okay. Then the definition, based on that one, we have defined enzymes. The definition of enzymes: soluble, colloidal, organic catalysts formed by living cells. As I said, specific in action, mostly protein in nature, except some ribozymes, inactive at 0 degrees centigrade and destroyed at 100 degrees. Okay. The second objective is classification. They are classified into 7 classes by the Enzyme Commission, by International Union of Biochemistry: EC1, EC2, EC3, EC4, EC5, EC6, and EC7. EC1 is oxidoreductases, EC2 is transferases, EC3 is hydrolases, EC4 ligases, EC5 isomerases, EC6 ligases, EC7 translocases. The first one we discussed, EC1, oxidoreductase, carrying the oxidation reduction reactions. Okay, then here reduced and then oxidized. Again, you can see here there is reduction of the B and substrate and oxidation of A substrate. So we have taken the lactate as an example and explained how the lactate is converted to pyruvate, reducing the NAD into NADH. There are a number of subclasses are there. There are dehydrogenases, reductases, oxidases, peroxidases. So under the subclasses, there are some of the examples are lactate dehydrogenase, glucose 6-phosphate dehydrogenase, peroxidase, cytochrome oxidase, and malate dehydrogenase, and many other enzymes. The second one is transferases, EC2. Here I have shown that how the transfer is taking place. Transfer of C, N, or the P or phosphate or methyl group. So here serine, we have taken the serine as an example, and then serine and with the reaction with the tetrahydrofolate by the action of serine hydroxymethyl. So transferase, so leading to the formation of glycine, and so N5, N10 methylene tetrahydrofolate. Okay, then there are subclasses: aminotransferases or transaminases, kinases, AST, ALT, ornithine transcarbamylase. So like that. Okay, so there are many examples are there like hexokinase and creatine kinase. Okay, then the third one is hydrolases. So here, in the presence of H2O, the breaking of the bond is taking place. The example is given here: urea, end product of amino acid catabolism, leads to carbon dioxide and ammonia. The examples are acid phosphatase, enzyme examples: lipase, amylase, trypsin, lactase, sucrase, and alkaline phosphatase. The fourth one is EC4, Ligases. It is also breaking of the bonds taking place, but without the requirement of water. CC bonds and carbon-sulfur bonds, carbon-nitrogen bonds are broken. Here I have shown how the breaking of the bonds is taking place, leaving behind double bond. Then, here the example is pyruvate is converted to acetaldehyde and by the action of pyruvate decarboxylase. Then, here the examples are aldolase and fumarase and arginosuccinase and carbonic anhydrase and decarboxylase. Okay. The fifth one is isomerases, catalyzed rearrangement of optical and geometric isomers. Okay. Here I have shown how it takes place. The example which I have taken is methylaminal coenzyme A by the action of mutase leads to succinyl coenzyme A. So phosphoglucomutase. Okay. Then phosphoisomerase, epimerase is another example, and retinol isomerase is one more example. The sixth one is ligases. Here catalyze the formation of bonds between carbon oxygen and carbon sulfate, carbon nitrogen, also carbon carbon. Okay. So here the ATP is used and also GTP is used. Okay, this is coupling with the ATP and then leads to formation of bonding and then ATP is utilized here. Here you can see how the carbon-carbon bond has taken place. Here you can see here. Okay, so here pyruvate combining with the CO2 by the action of pyruvate carboxylase enzyme is pyruvate carboxylase. Pyruvate carboxylase enzyme leading to the formation of oxaloacetate. Okay, the examples are glutamin synthetase, as I said, pyruvate carboxylase, DNA ligase, one of the most important enzymes in the DNA replication. The final one is the translocases. It transfers the ions across the membrane or they are molecules, okay, across the membrane. So their separation, either the separation within the membrane, also they carry out. So what they translocate? So the translocate like hydrons, okay, the inorganic cations, anions, peptides and carbohydrates, amino acids. So these are various aspects of the different classes of enzymes and definition of enzymes. In the next session, we'll take up some other topic about the enzymes. Okay, thank you very much.